Bismillahirrahmanirrahim. Assalamu alaikum, dear student. My name is Dr. Muhammad Adnan Asgar, and today this would be the last lecture of the characterization of pharmaceutical materials by using thermal techniques. So far, we have studied some of the important aspects of characterization of pharmaceutical materials by using diffraction scanning, calorimetric, and sometimes thermogravimetric analysis coupled with TTA. In this lecture, we are going to study the protein denaturation. Protein denaturation is actually a journal term used to describe a change in the structure of a protein. This change usually results in a non reversible unfolding of the protein that affects its shape and therefore its biological activity. There are various techniques that could be used to measure protein denaturation, including changes in physical properties, which means the solubility, and the changes in reactivity, such as with enzymatic proteins. With diffraction scanning calorimetry, protein denaturation is actually the measurement of thermal stability of a protein in the solution. A low energy endothermic peak is observed over a temperature range. The temperature of the peak provides us some information about thermal stability, and the area of the peak is a quantitative measure of energy absorbed by the protein in order to change the structure. Experimental conditions such as heating rate, pH, ionic strength, and even protein concentration can affect results. In order to minimize the opportunity for aggregation of protein, most measurements may be performed at relatively low concentration. Relatively low concentration means 1% or 10%, 10 mg per ml of the solution. Diffraction scanning calorimetry for the denaturation of albumin from chicken eggs is shown in this picture, which is actually the comparison of 1% concentration as well as 10% concentration, which is quite obvious in this diagram. At 1% concentration, a peak of about 40 microvolt in height and 0.21 joule per gram in area is easily detected. The sharpness of the minor detail associated with peak is however less clear because of low signal strength. At 10% concentration, the peak is about 400 microwatt in size, and there is excellent detail showing minor shoulders on both the low and high temperature sides of the peak. The fact that peak temperatures differ by only 0.2% degree Celsius which provides confidence that minimal aggression occurs at 10% concentration. This is great flexibility in analyzing the data. For example, in this picture, the 10% concentration data is shown with sigmoidal baseline, and the present denaturation is plotted as a function of temperature. A plot of time during experiment shows that the entire experiment may took only 1 hour or 60 minutes to complete, which is quite faster than typical obtained with microcalorimeters or solution calorimeters. Here is the freeze drying. Freeze drying or lyophilization has become a standard process in the pharmaceutical industry for the manufacture of biologically active substances. However, it is not without limitation due to its high cost in capital and energy, long processing time, and difficulty in selecting manufacturing conditions of time, the time, temperature, vacuum, and component concentration. All of these parameters must be optimized in order to achieve a final product with the desirable characteristics of full activity of the protein or drug, easy reconstruction, acceptable appearance of freeze dried cake, good storage stability. So these should be kept in mind while obtained to obtain optimized in order to achieve final product with desirable characteristics. The process of freeze drying relies on the vapor pressure of ice. Even at temperatures as low as minus 50 degrees Celsius, ice sublimes and leaves a very porous, low density cake containing the stabilized drug. Since the sublimation rate, which is actually the drying rate, is very temperature dependent, use of highest possible temperature during primary drying provides maximum drying efficiency and lowest process cost. In order to select the optimum drying temperature, it is necessary to understand the physical characteristics of the components used in the formulation that is to be freeze dried in decreasing order of mass. These are typically water, bulking agents, buffers, or stabilize, and finally the drug itself. The bulking agent, which can be either crystalline or amorphous, and its interaction with frozen and unfrozen water in frozen solution. It actually defines the physical structure which is essential to the successful freezing. This structure manifests itself in the form of transitions that occur at specific temperatures. Physical properties of bulking agent such as modulus or viscosity can change by orders of magnitude depending on whether the process temperature is a few degree above or below the transition temperature. The transition temperature sometimes is also known as the glass transition temperature. Therefore, knowledge of this structure and how it changes with time and temperature is required for successful drying. DSE has been used with only at modest success in characterization of frozen solutions used for freeze drying. The reason is that there are numerous components and transitions happening within a narrow temperature range, and diffraction scanning calorimetry can only measure the sum of these. In addition to this, diffraction scanning calorimetry must use relatively high heating rates, 10 to 20 degrees, in order to optimize sensitivity, while the best resolution is obtained at low heating rates of zero point 0.5 to 1 degree Celsius per minute. In the introduction section of new diffraction scanning calorimetric technologies, it was explained how modulated diffraction scanning calorimetry has both an advantage and on average and modulated heating rate. The combination of two heating rates allows the operator to select a slow average heating rate in order to obtain good resolution and a higher modulated heating rate to obtain increased sensitivity during the whole experiment. In addition to this, resulting modulating heat flow can be separated into heat capacity and kinetic components of the total heat flow in order to improve ease of data interpretations. This figure actually shows the raw modulated heating rate and modulated heat flow signals from an mm, modulated diffraction scanning calorimetry. This experiment actually performed at an average heating rate of zero point 0.5 degrees Celsius per minute on a frozen solution of 40% sucrose in water. The change in heating rate, which is the modulated one, is approximately 3 degrees Celsius per minute, which causes the modulation in the heat flow signal. This figure is actually shows that the calculated signals from this modulated diffraction scanning calorimetric experiment. The total signal is very difficult to interpret because it is equivalent to standard diffraction scanning calorimetry at the same heating rate and contains two different transitions, and we were very quite familiar with the differences between the modulated diffraction scanning calorimetry and the conventional diffraction scanning calorimetry. The sum of the important differences are the priorities of modulated DSC over the conventional DSC was actually the the purity of the compound, and it have sharp temperature measurements in a wide temperature range. Moreover, it gives flat and reproducible baseline and increased sensitivity with increased resolution, and it would be quite helpful for the separation of complex transitions, more accurate measurement of crystallinity in semi-crystalline materials, and the direct measurement of heat capacity while programming temperature or holding it isothermal. The total signal is quite difficult to interpret because it is equivalent to standard diffraction scanning calorimetry at the same heating and contains two different transitions. The first of these is the important glass transition seen in the reversing signal between minus 43 degrees Celsius and minus 39 degrees Celsius, which is quite obvious in this diagram. Here we can see this is quite obvious and can be observed by using modulated diffraction scanning calorimetry instead of using conventional DSC. The second transition is actually an exothermic in the non-reversing signal caused by crystallization of free water that could not crystallize during the quench cooling of the sample. This peak shows a maximum at about minus 36 degrees Celsius, which is quite obvious in this diagram, and heat of crystallization is about 5.7 joule per gram, which is written in this picture. It is not surprising that unfrozen water would begin to crystallize near minus 42 degrees Celsius, while since this is near the onset of the glass transition where a significant increase in molecular mobility and diffusion can occur. So this is actually the advantage of modulated diffraction scanning calorimetry over the conventional DSC. If we performed this experiment uh, with the help of conventional diffraction scanning calorimetry, then the results would be all different because conventional diffraction scanning calorimetry usually do not give sharp change, do not observe the sharp change or the peaks in a close temperature proximity. It is unable to identify, it is unable to record the clear message obtained during the enthalpy change of any sample. The accuracy of conventional diffraction scanning calorimetry is not quite good because the temperature range in some samples are very close. The Curie temperature of the samples are quite close, so it is that conventional diffraction scanning calorimetry is quite unable to differentiate between the glass temperature or the Curie temperature, which are very adjacent to each other. In this case, the modulated diffraction scanning calorimetry play a vital role for the identification of different phase transitions occur in a compound in a close proximity of the temperature. This happens in this case where we can see that heat flow is observed while reversing this experiment from minus 60 degrees Celsius to minus 20 degrees Celsius. While reversing this sample from minus 60 to minus 20, there is an observation of some enthalpy change at about minus 43.66 degrees Celsius. Just passing through this temperature, there is a heat anomaly and it is recorded by modulated diffraction scanning calorimetry in a quite easy way. While at about minus 39.41 degrees Celsius, there is an other enthalpy change which is recorded quite successfully by modulated diffraction scanning calorimetry. So here is the temperature difference of about 4 degrees Celsius, which is very close to each other. So this close, even at the close temperature range, modulated diffraction scanning calorimetry actually observe and record the good results of heat enthalpy while reversing the heat flow from minus 60 to minus 20 degrees Celsius. The same is observed while non-reversing heat flow. The observed enthalpy changes are quite close and will and recorded by modulated diffraction scanning calorimetry successfully. This figure actually shows the modulated diffraction scanning calorimetric data obtained on the same sample as we discussed earlier, except that it was cooled at zero point 0.5 degrees Celsius per minute as compared to the quench cooling that was used in previous run. The cooling and heating data are actually plotted in heat capacity unit so that they can be visually compared. Notice that the slow cooling produces a more complex structure which has two-step changes in heat capacity. The derivative signal more clearly shows that the double transitions, and it actually shows that there are very minor temperature lags between heating and cooling with T0 diffraction scanning calorimetric technology. Even at slow average heating rate of zero point 0.5 degrees Celsius per minute, modulated diffraction scanning calorimetry provides extremely high sensitivity for characterizing the complex structure of frozen solutions which are used for freeze drying. This is all about the uh, pharmaceutical characterization of drugs by using thermogravimetric analysis. The most important thermogravimetric analysis technique that we studied for the characterizing of pharmaceutical material is the diffraction scanning calorimetry, especially the modulated one, along with the thermogravimetric analysis. For this lectures, you people should consult the book named Thermal Analysis Fundamentals and Applications to Material Characterization by Rahman, while another scientific paper uh, with title Characterization of Pharmaceutical Materials by Using Thermal Analysis by Thomas. These book and this paper should be consulted, and this is all about the characterization of pharmaceutical materials by using thermal analysis. Thank you very much.